Hello. Hello, everybody. I'm Ditlev, and I want to present you some recent work from the Center for Fundamental Living Technology at the University of Southern Denmark. We have developed a closed-loop system that is capable of optimizing complex systems. Let's move on. Yeah. So here we see the three components of the system. On your right-hand side, we have the simulation part. This is the simulation that carries out the complex system in simulation. And in between, we have a message system, which takes the results from the simulation, the results we call responses, and passes them to an analysis framework, which is an artificial intelligence design of experiments-based neural network, which is capable of predicting experiments that are more likely to increase the response. So these experiments are in this loop passed on to the messaging system, which then triggers a set of simulations. And this just carries on until you have reached the desired result, or you complete a set amount of or a fixed amount of iterations. So this artificial life design of experiments-based system, also called prediction design technology, is something that comes from Daptics. They provide an application interface layer for us to connect to. So instead of a lab, we have we perform the experiments in silico. And Daptics models it builds a new model and predicts these experiments that are likely to be better. So for each iteration, the algorithm explores the experimental space of the complex system. In practice, this is how it looks like. We have the command line interface. Initially, we have some configuration. And over on the right-hand side, you see a configuration file in JSON, where you can set what kind of engine you want to simulate in. Is it MATLAB? Is it Python? And how many generations, also iterations, you could call it, do you want to run? And how many simulations do you want to execute for each iteration? In the configuration, we also have the experimental space definition, which is a set of parameters and the type of set parameter and the values that it can take. So after the configuration and initialization, it begins with the first generation, calculates the responses for all of the set of simulations, saves it locally, and uploads it to the analytical framework. And then the framework comes with a new set of experimental parameters for which we can carry out a new set of simulations. And this goes on until it finishes. What system have we optimized using this system? It's based on it's a model of a protocell that was originally developed at Los Alamos National Laboratory, and in the literature it's called the Los Alamos bug, but now it's also known as the Flint protocell. So Steen Rasmussen gave a talk on Thursday about the system in detail, and here we are trying to optimize a subsystem in this protocellular model, which is the information system. So just to give a quick recap of this system, we have a replication here, a replication process, and initially we have this DNA strand, which is anchored to the surface of the protocell with a hydrophobic end, and we introduce two complementary oligomers that will hybridize to this DNA strand. And after the hybridization, they are ligated together, so they connect. And thereafter, they will dehybridize, or rather dissociate, and allow for a new replication iteration to occur. And this is another representation of the system. It's simplified as we don't take account for the anchors to the surface of the protocellular model. And we have also shown here the different reaction constants that are within the system. And these are interesting to us because if we can control these reaction constants, we can also control how fast will the replication occur. So specifically, we have the ligation rate here, and we have the dissociation reaction constant over here. And these are some we would have an increased interest in. Steen also presented a system by Julian Gibbs Davis and others called Lesion Induced DNA Amplification, or LIDA. And this allows us to control the reaction constants in a sense. We can introduce new components like an A base, which is basically just a nucleotide without the base. And that is more destabilizing than a regular nucleotide match. We can also introduce a mismatch to gain a fine control of the stabilization. So how does the off rate or the reaction constant K minus relate to the thermodynamics? We have found that the on rate is actually pretty constant throughout the composition, or if you change the composition of the nucleotide strand, the on rate is pretty constant. So we're interested in the off rate here, and we could calculate the off rate if we have if we know the change in Gibbs free energy. And the change in Gibbs free energy is something we could calculate. And on the right hand side, we have the three types of components that we could introduce. Up here we have the match, which is quite stabilizing. We have a mismatch, we have the A base, and we also have a bulge. A bulge is where you introduce more nucleotides than what the complementary strand has. And this can result in the formation of some bulges and also some stem loops, which can increase or increase the stabilization of this duplex. So we have modeled the system. We have the change in concentrations of the various chemical species over time. And I won't go too much into detail about this because I want to move on to the more interesting part, the actual optimization. We have defined a set of parameter intervals for these reaction constants, and we have entered them into this round of optimization with a certain objective. We wanted the amplification to reach at least 90 micromolar. And if this is achieved, we capture the time at which it is achieved and send this off to the prediction design technology. And here we have a sorted bar plot sequence with the generations or the iterations on the x-axis, and on the y-axis we have the response, which is time, a negative time actually. So minus 60 is actually 1 hour. So we had some simulations that performed really poorly, but we kept them to 1 hour because otherwise we wouldn't be able to see how the faster simulations performed in this graph. So we see that the optimization from the first generation to the second generation quickly finds quite fast amplification experiments, and it stays that way for quite a long time throughout the next generations, and then it starts to go into a more exploratory role where it starts to seek out new areas in the experimental space where there could be another optimum, or perhaps the global optimum if it hasn't been found yet. So we started out with a very fast ligation rate of 10 to the minus first per second. And here we have a regression tree that shows the main explanatory rule for the system, and this is the off rate for the dehybridization or the dissociation of the duplex, and that is unrealistic because we cannot find an enzyme or anything like that, or a compound that could actually give us this kind of fast ligation rate. So we had to do a new round of optimization, and with a more realistic ligation rate. And here we found that instead of the dehybridization being the inhibitory effect in this system, we now see that the main explanatory rule is the ligation rate. So this was really interesting, and it's also something that Steen Rasmussen and others have found manually before. So it was a confirmation of this optimization loop. And here's another graph just to show you how it relates to the experimental data from Julianne Gibbs-Davis and the others, the work that they have done. We have plotted it here in blue. The blue dots are the experimental data. And we have tried to set up some parameters according to this experimental data. And with our model, we built the green curve here, which approximates the experimental data pretty well. The black curve is the optimization, where we used an unrealistic ligation rate, and the red one is the realistic one. So we have found something that is even faster than what was shown experimentally. So to recap, we have developed a closed loop system that is capable of optimizing complex systems autonomously in simulation. We have optimized the LIDA system, found better parameter combinations that yields faster replication rates. And also, it's a confirmation of the LIDA system for us to go forward with that. For future perspectives, we would actually like to set this up in a laboratory setting. So instead of having in silico simulations, we could actually have real experiments carried out autonomously. That's really interesting. And we would also like to apply this optimization framework for the optimization of the other two subsystems, namely the metabolism and the container. And we also talked about going open source, because this tool could really be, we're interested in seeing how other people could use this tool to optimize certain processes. But we are still a bit far away from that. So it's some work in progress. So I want to acknowledge Daptics, who have been a great support throughout this research work. And I also want to thank Christina Sørensen, who has done a lot of testing and also contributed to the model that we have used. And lastly, Steen Rasmussen for spearheading this project. So thank you all for listening. And if you have a question, you can ask it right away, or find me on Slack.
Okay, thank you so much. So we do have time for one quick question if somebody has one. All right. Well, of course, as you know, you can go ahead and ask your questions on Slack if you think of something later. Besides that, we'll now move back over to Lecture Hall A for the morning's keynote. So, thanks, everyone.